Carbon is you'll hear it say like we are carbon-based life forms. Carbon is the main element that makes up living organisms, and the reason is because carbon is so good at bonding with other atoms, and that's going to allow carbon to form these very large, complex, and just varied shaped molecules that we'll begin to go over. And you'll hear about a class called organic chemistry in college. Maybe if you got some older siblings or friends, they'll say a bunch of bad words about organic chemistry. Organic chemistry is the study of organic compounds. What's an organic compound? It's basically anything that has carbon in it. So organic chemistry is really studying a lot of like the molecules that make up living things. So there you go. Moving on. Get my cursor over here. The four main biomolecules, that's kind of the verbiage you heard from freshman bio, are carbohydrates, lipids, proteins, and nucleic acids. So carbon makes up all four of these. And each of these molecules has different roles in the body, and we'll learn about those throughout the year. But the goal of Unit 1 is that you can identify, if I show you the structure of a lipid, you can say that's a lipid, not a protein, stuff like that. And you know like the basic atoms that make them up, and so on. So that's kind of what we're building towards. And these are also called macromolecules, so bigger molecules. Macro means big. Carbon makes up life. Moving on. Now, I said carbon can form all different kinds of molecules. So let's break down, kind of reviewing some basic chemistry, what's going on there. Remember electron configuration, like 1s2, 2s2, that kind of stuff? Electron configuration is basically talking about the placement of electrons in the atom. Now, when we're trying to understand how does an atom behave with other atoms, it's all about the valence electrons. Valence electrons, think of a veil. It's on the outside of like the bride's face. Valence electrons are the outermost electrons. So if you look at hydrogen, you remember the Bohr model, like the rings and stuff? We say hydrogen has one valence electron. We say oxygen, as I'm looking at the slide, they don't have the valence electrons here. We'll say VE. Hydrogen's got one valence electron. Oxygen's got 6. Nitrogen has 5. Carbon has 4. The numbering on our periodic table up here, it's in Sharpie, like 1. Everything in this column, they got one valence electron. 2. Remember the transition metals, like that valley? Those are the weird ones. They have different numbers of valence electrons. They can recruit from their d orbitals. Everything here has 3, 4, and so on, kind of coming back together. Now, the valence electrons is related to the number of bonds something can form. The number of bonds atoms can form is related to the number of electrons they need to fill the valence shell. I should have mentioned last class that atoms behave so that they get a full outside shell of electrons. They want a full valence. That's when atoms are stable. Atoms don't want to have empty valence shells. Okay, so if you're hydrogen, hydrogen needs just one more electron to have a full first shell. So that's why you see the purple little outline there. That's saying, hey, hydrogen wants one more electron. So anytime you see hydrogen, you'll notice hydrogen forming one bond with something else. Oxygen has a need for 2 more electrons to get that full valence shell. So oxygen typically forms 2 other bonds. Nitrogen has 5 valence electrons. It wants to gain 3 more to get that 8. So it'll form 3 bonds. Basically, atoms want 8 valence electrons. They call that the, remember what it's called? What rule?
Octet.
Octet, yeah, good. The octet rule. Okay. Carbon wants to form 4 bonds. It needs 4 more valence electrons. And this in lies why carbon is so important. So make sure you get that. Carbon is so important. It's the main, we're carbon-based life forms because carbon can form 4 other bonds. It's got a lot of flexibility, right? Okay. Let me write up an example here on the board. So like carbon dioxide, you'll see it drawn like this. Why it looks like this is carbon can form 4 bonds. So it's only paired up with 2 other atoms, but you'll see that the 2 lines, remember it's a double bond. So there's 2 bonds there. Oxygen only needs to form 2 more bonds because it needs 2 more valence electrons. That's why there's only 1 oxygen connected to 1 carbon. Okay. Moving on with it. Oh, here's your so carbon dioxide. What's interesting about this picture is they showed it. They did basically a different way to show a double bond. They show a double bond here, how it would look in the Bohr model. So remember, in a bond, you're sharing a pair of electrons. So that right there, that pair of electrons being shared, that represents 1 single bond. And then this would be another single bond, and collectively you call that a double bond. So this would be like a line, that would be like a line. Okay. If you have questions, by all means, raise your hand. Or let's keep going. Okay. And then same thing over here. Cool. All right. Here's just another example, just trying to show the different things carbon can form, right? Carbon will tend to form these long chains with itself. Especially you'll see this with your lipids. You'll get these long, they call them hydrocarbon chains. I'll write that word out. Hydrocarbon. Hydro sounds a lot like hydrogen. Carbon, carbon. If you're at hydrocarbons, oil and gas industry. Hydrocarbons make up like our fossil fuels and stuff. So just making that connection. But yeah, again, what you'll notice is carbon is forming 1, 2, 3, 4 bonds. This carbon's got 1, 2, 3, 4 bonds. Hydrogen, notice how hydrogen's only forming 1 bond. So again, it all goes back to the number of valence electrons and how many more valence electrons they need to fill that valence shell. Okay. So this is just kind of showing, just sort of introducing you to the different ways that you can see formulas written out. So like, for example, a molecular formula, right, a little bit bigger, like CH4, bless you. That shows a molecule that has 4 hydrogens and 1 carbon. If you write that out just as a structural formula, not trying to show it in 3 dimensions, you would do it like this. But then if you remember it from chemistry, you learn about the shapes of molecules and stuff, like tetrahedral, trigonal pyramid, bent, stuff like that. That's what they're trying to show here. You don't have to know all those shapes for AP Bio. This is more just trying to get like quick refresher of some basic chemistry before I start showing you the different like biomolecules and stuff. Okay. Ethane, it's where you'll have 2 carbons next to each other. If you take organic chemistry, part of organic chemistry is they make you memorize like CH4, what it looks like. Ane means it's just all single bonds. You don't have to know this, you're just kind of curious. Just some beginning organic chemistry. Ene's referring to double bonds. So that's where you see a double bond right there with the carbon. Okay. Make sure I said everything here. I talked about carbon dioxide. Okay, you're good. Make sure I said everything here. Yeah, an example of a tetrahedral shape would be like what you would see with methane. So with methane, methane CH4. You have that memorized from chemistry? One of those you gotta memorize. So you have 4 other four atoms attached to the center atom, so you would call that tetrahedral. Tetra means 4. Anytime you have a double bond, if I go back to this picture, so anytime you have the double bond, everything is in the same plane. So it's flat as the molecule. Okay. And then I talked about all this. Valence, that's just telling you how many covalent bonds something can form. So I think I mentioned last class, a lot of times I'll be teaching from the pictures and I'll go through here just make sure I said all the bullet points here. So these are more helpful maybe if you're reviewing the PowerPoint slides, you can kind of go through, read those bullet points. Okay, now let's get into some of the different structures carbon can form. So carbon wants to form these carbon skeletons I mentioned earlier, where you get basically a bunch of carbon atoms linking together in these chains, these hydrocarbon chains, right? And the chain can vary in lengths. So if the chain is made of 2 carbons and there's all single bonds, they call it ethane. If there's 3 carbons in a chain, You could you call it propane, right? King of the Hill. Is King of the Hill still like part of? You may know what I'm talking about. Say King of the Hill. Yeah, Bobby. Yeah. You know what I'm saying? Yeah. Anyways, propane. You know, grill and stuff. But then those chains can also branch. So, for example, butane, what's in like lighters, the hydrocarbon in lighters, is difference between butane and propane is just you have an extra carbon in that chain. So 4 carbons. If you take one of these carbons here and you just attach it right here, you take this carbon and link it right there, you have what's called 2-methylpropane. So this is part of like beginning of organic chemistry. Is like they make you like learn how to name all this stuff. You don't have to name it, but just real quick, the 2 is referring to where the chain is starting. It's starting at the 2nd carbon. You number the carbons going 1, 2, 3, that way. So the chain is coming, starting at the 2nd carbon. Methyl is referring to methane, right? Methane was CH4. So here's the methyl group. So 2-methyl, methyl is coming off the 2nd carbon. And then propane, because the chain is 3 carbons long. Propane is always 3 carbons. So anyways, again, AP exam's not going to ask you that, but just as you're seeing the name of these figures in your book and stuff, you kind of have a basic idea of how they're naming it. It's not like some complicated magic. It's actually a very systematic approach. Okay. Then we can have double bonds. And those double bonds can be put in different positions throughout that carbon chain. Where if we have a similar system I showed you on the last slide for where that double bond is located. So if the carbon chain is 4 carbons long, we call that butane, right? But then I said if you see ene's referring to the double bonds. And the double bond here starts at the number 1 carbon. Remember, you number from left to right. So 1 butene is saying the double bond is right here. 2 butene the double bond starts at the 2nd carbon. Okay? Then carbon can form these ring-like structures. Think glucose. If you've seen a picture of glucose, try to remember a picture of glucose. It's in these rings. A basic ring where it's like just only carbons and hydrogens, they call cyclohexane. It's like cyclo means circle. I don't know if cyclo means circle. That's what I just told myself. I don't know if that's actually true, but cyclo, we're kind of referring to like a circular type shape, let's say. And then hexane, like a hexagon, has got a 6 in it. So it's a ring with 6 carbons. Okay. Now, if you're wondering, like, what's this nonsense? Why is drawing like a hexagon here? This is like shorthand. It would take forever to draw all these hydrogens. So what this is saying is, hey, we can just assume that the vertici, what we call those, the corners there, we assume those are carbons, right? That's just everybody understands that. And then we all assume the number of hydrogens. Because carbon forms 4 bonds, you can assume that there's 2 hydrogens coming off there, right? Like when you get to that level of chemistry, it's like it's just assumed that you know that. Kind of like LOL. You don't need to like explain that to everybody, you know, like any mnemonic or acronym, right? Like we all assume that's what it means. Benzene is a similar ring-like structure, but in benzene, you'll get these 3 double bonds. And so then looking at the abbreviation, so they just show the double bonds where they're located. Any questions so far? Again, I'm not gonna like show you that structure, ask what that name is on the test. It's just trying to get you to begin to be a little familiar with how people start naming a lot of these structures. Yep.
Why are in the first one 2 hydrogens to a carbon, and in the second one it's only 1 hydrogen?
Oh, like, are you wondering?
No, in the second one, in the presence of rings, like the pictures.
Oh, okay, I gotcha. So you're asking, let me get it clean. You're like, okay, here there's like 2 hydrogens coming off it, but over here there's only 1. Oh, good question. Remember, hydrogen can only carbon can only form 4 bonds. 2 of those bonds are being used up for the double bond. Another of those bonds is being used to link up with another carbon. So there's only 1 more bond left. So you can only have 1 hydrogen.
Okay.
Whereas in the cyclohexane, where there's no double bonds, because you're not using up a bond for a double bond, you now have the space to form 2 other bonds. That's actually a really important detail. Make sure you understand that. Right? Want me to explain that again? So there's more hydrogens here because you're not using up the bonds for carbon in a double bond. Or carbon can get 4 bonds. Okay. All right, let me make sure I set everything here, these next few slides. Remember, organic molecules, something made of carbon. Hydrocarbon, it's just made of carbons and hydrogens. Hydrocarbons, they make up our fats, our lipids. And yeah, hydrocarbons release a lot of energy. That's why we put them in our cars and stuff, right? Or our lighters or our propane tank, stuff like that. When you break those carbon to carbon and carbon to hydrogen bonds, you release a lot of energy. All right, so I want to talk about the different ways we can arrange these molecules. So let's say you have the formula like, let's go even simpler than that. Let's say you're looking for a mate, right? You're looking for somebody to date, and you're like, I'm looking for somebody in finance. 6'5", blue eyes. There's a lot of people like that, right? That's just a generic category. But like, you found that one person who's like that. You found that one person. What I'm getting at here is you could have a molecule with this would be 1, 2, 3, 4, 5, 6, 7. This would be C3H7F. That is the chemical formula for this. Okay? 6'5", finance, blue eyes, all that. But the way you arrange those atoms can vary. That's what we call an isomer. Isomer, same chemical formula, but how you arrange those atoms varies. Okay? So that's something, I don't know, that blew my mind the first time I learned that, like in organic chemistry. Like, you're usually hearing chemical formulas, but there's many ways you can arrange those molecules. Okay. Now, a structural isomer is just saying, this is the fluorine atom. It's saying, hey, the fluorine is just being put at a different spot. Same number of atoms, fluorine's in a different spot. You don't have to know like all these other ones. I'll come back to this slide, but it's just trying to like show you the different kinds of isomers. So don't pass out on me. I promise you it's not this crazy. But just same structure, but they're arranged in different ways. Okay. Okay. So if we start with the first one, a structural isomer. Structural isomer is this first one. Okay, so it's what I just said. Another example they gave you is 1-bromopropane. There's also 2-bromopropane. 1-bromopropane, the bromine, bromo, talking about bromine, comes off the first carbon, right? 1, 2, 3. Propane was 3-carbon chain, so 1-bromopropane. But the chemical formula is this, C3H7Br, right? Now there's 2-bromopropane where the bromine is now coming off the number 2 carbon. But the chemical formula is still C3H7Br. See what I'm saying there? So same formula, you just attach them in different ways. Okay? All right, so that's this side. So now I want to talk about stereoisomers. Sometimes you'll hear them called spatial isomers. And there's a bunch of different kinds, but what I want to talk about is this next slide is this one specifically, the cis-trans isomers. Part of a category called diasteromers, easy for you to say, but you don't need to know like conformers or rotamers and stuff. This is the one that's important for us. We'll also talk about enantiomers here in a minute, but these are going to be the two that you're going to have to know. Okay, these other two, don't worry about. So let's talk about it. So what's going on here is, I mentioned you got the same chemical formula. You can change where you place these things, but what can happen is in 3-dimensional space, where these chlorines are located can switch. So if the 2 chlorines in this one are on the same side, we say it's cis. But if the chlorines are on opposite sides of this carbon-carbon double bond, that's your anchoring point. So the chlorines are on the same side of the carbon-carbon double bond, cis. If they're on opposite sides of the carbon-carbon double bond, it's trans. The naming, especially with like, you know, how people say cis trans, it actually makes sense with how they name it in the molecules. That makes sense. Question?
Can it be possible for those two chlorine atoms to be only on the right carbon?
Like you're saying, like this,
Yeah.
and then like that.
Yeah.
Probably. I don't see why not. But
Or would it make it too forward?
it would definitely behave much differently. That's for sure. But to be honest with you, that's beyond my chemistry knowledge. I would say for our purposes, I just need you to know cis and trans, right? Just knowing like where they're located. But I like where your mind's going. Appreciate the question. They say inflexibility of double bonds. So double bonds, because they're so strong, those bonds are very rigid. So what can happen here is basically this double bond, like these groups can rotate. So like this chlorine can like rotate down, and same with that chlorine. So some molecules they can swap between cis and trans, it's almost like on a hinge. If you see what I'm kind of trying to say there. Like it'll do this. Like that double bond can rotate, can flip, swapping from cis and trans. Okay. Now, you're wondering like who cares? Well, how a molecule behaves, whether it's in a cis formation or trans, makes a massive difference, especially like in the medical field where they're making medicines. Anyways, I'll show you an example in a couple slides, but yeah, whether a molecule is in the cis or the trans conformation can make a big difference in how it behaves. So just take my word for it for now. Okay, now let me back up to that this slide here. So we went over cis and trans. I want to show you now the enantiomers now. Okay, so these ones right here. What's going on with these is basically enantiomers are mirror images of each other. So molecules can rotate. You gotta remember, molecules exist in 3 dimensions, right? And they can have these spins. When light hits them, they can spin either like clockwise or sometimes counterclockwise. So what's going on here is, if you look at this molecule, all of the groups attached to the carbon are the exact same, right? You got an OH, you got a hydrogen, CH3, COOH, and their relationship to each other are all in the same. Like the OH and the H are next to each other. COOH is on top of that. But enantiomers, they're just mirror images of each other. So it's interesting. Like everything is attached the right way, but it's like your hands. Like, they're the same, but they're not. They're mirror images of each other, right? But like the position. My middle finger is the middle finger is what I'm trying to say, right? The middle finger is like it's not. They're not like the middle finger isn't over here on this one and over there, right? It's the groups are all in the same location. Okay. Anyways, and that makes a big difference with how molecules behave. So another example, like a medical one, would be ibuprofen. So whether or not ibuprofen is the left hand or right hand, the left hand, right hand is referring to like how it spins, whether it's going this way or say that way. So if we come over here, if you have the plus means it's the right hand, minus means left hand. You don't have to have that memorized. I'm just trying to just try to introduce you to just the basic structures these molecules can take. Whether or not it's spinning to the right or to the left, that changes how effective the ibuprofen is. Racemic, when they say racemic mixture, is saying it's just a 50-50 balance of left and right. But the right-hand one is the most effective form of the ibuprofen. Okay. How are we doing so far? I know it might be kind of a lot. Okay. And they're non-superimposable, just like your hands, right? They don't overlap. So if you take this and put it on it, this group is over here, but that group is over there. Okay. Now, getting into what are called functional groups. So what you'll see in biology a lot is you'll have the same basic template or blueprint, but then like molecules can change what they call the functional groups. It would be like syllabi. Seen a lot of syllabi this week. A lot of them have the same basic structure and layout, but different teachers change different sections to show their course. You'll have that happening in biology, where you have estradiol makes estrogen. You might have heard of it. Testosterone, heard of that before. Well, the two molecules have the same basic template. That all these different ring shapes, right? The benzene ring, the cyclohexene ring. But testosterone will have a double bonded oxygen group, whereas estradiol, you just see this OH group. And then testosterone has an extra CH3 attached there. And the point to get out of this is these molecules behave super differently in the body, right? Like we're talking like maleness versus femaleness, right? With like testosterone versus estrogen. So just small changes like that can make a huge difference in the body. Okay. All right. So let me make sure I said everything here. Oh, what is a functional group? Basically, you'll have these carbon chains where the basic template, right, the default is all these hydrogens. But a functional group is when you replace a hydrogen with something else. So you swap it out, you customize it. Is that what they call a mod? A modification. Functional groups are like modifications to your template. Okay? So there's a bunch of different functional groups that are important in biology. There's 7 of them. I'm going to show you here in a second. But they all behave in slightly different ways when they're doing chemical reactions. Okay, these are the 7 functional groups, the 7 ways you can kind of modify these carbon chains. Hydroxyl group, methyl group, carbonyl, carboxyl, amynyl, phosphate, and sulfhydryl. So I would want you to be able to like recognize the difference between these. You don't have to be able to draw a phosphate group, but if I show you a phosphate group, I need you to be able to say that's a phosphate group, not a methyl group. That makes sense. There's a little bit of difference in like level, you gotta know it. You do organic chemistry, you gotta be able to draw these. For right now, just recognize them. So let's go through them. First one, hydroxyl group. And the names kind of give them away. Hydro sounds like hydrogen. Oxyl sounds like oxygen. So hydroxyl is an OH group. It's also called an alcohol. You may have heard of ethanol. It's in a lot of alcoholic beverages, Ethanol, where it gets its name from is ethane. Ethane is a 2-carbon chain. But then if you swap out, this was a hydrogen, right? Default, the stock was all hydrogens, but you swap out one of the ending hydrogens with the alcohol group, the OH group, and we call it ethanol. I got a question. Oh, well, now I got a question for you. What do you think this would be called? Anybody? Yeah. Propanol, yeah, good. In other words, the OL, that's the ending that they always will switch it to. Propane, that was a 3-carbon chain, but the propanol would then just be you swap out a hydrogen with the OH. Okay, making some sense? All right, then you have a carbonyl group. So name kind of gives it away. Carbon, thinking of carbon, okay, obviously. But then what's maybe not as obvious is it has a double bonded oxygen. And then what's going on with this down here is that there can be other things that come off of it. So anytime in a molecule you see a carbon to a double bond of an oxygen, that's a carbonyl group. Meaning you could have this one right here where it's in the middle of the molecule. We call that a ketone. So ketones are in the very middle of a whole chain. Or you could have something called an aldehyde. Aldehyde is at the very end of a molecule. Where is the hide on a horse? The end of the horse, I think. All right. People on horses around here? I think the hide is like the end of the aldehyde is a carbonyl group, but it's at the very end of that chain. Okay? I don't really care if you know ketone versus aldehyde, but you do need to be able to recognize that carbonyl group. The carbon double bonded to an oxygen. Okay. All right. Going on to the other ones, you have a carboxyl group. So sounds kind of like a carbonyl group, and that's because it's actually very similar, right? You'll see that C double bond to an O that was in a carbonyl group, but the carboxyl group, the difference is you now have an alcohol. So that's kind of where that the L, the very end of it's coming from. So a carboxyl group, you could have acetic acid. This is vinegar, where you have there's a carboxyl group. Don't worry about the ionized stuff. That's not that big of a deal for us, for our purposes. But basically, yeah, you can lose a hydrogen, and so that's why they call it. Remember, acids lose hydrogens. Acids would give up that H. Not super important for this conversation though. Amino group, that one is NH2. So you have an N going to two H's. Oh, make sure you can tell these groups by like the symbol, the chemical formula there. Not just what they look like, but if I say COOH, that you know that's also carboxyl. Okay. Some of them it's not as tricky, but like just, you know, slightly different skill than just visually seeing it. Okay. All right, amino group is NH2. I remember the amino because it has the N in it. So N is the only one that's got the nitrogen in it. And again, don't worry so much about the this whole the ionization thing. Not quite as important for us right now. Yep.
Is it because of it's basic, so it accepts the H?
Yes, but you don't have to worry about so much that right now. I just want you to get like the basic names of it. But yeah, good. Okay, then the last three, sulfhedryl. This is also called a thiol group. Sulfhedryl has the SH. So, and that makes some sense, right? Sulfur, hedryl, hydrogen, and then they'll also call that a thiol group. So that one can be a little tricky, but you still, I don't know, like the sound, like when you have the H sound there, I don't know. It doesn't seem like they quite go super well together, but I always found it wasn't too bad to notice the difference between them. An example of a molecule that have the thiol group is the amino acid cysteine. So you can see that SH group right there. Okay. Almost there, a phosphate group. That one should be easy, right? Polyatomic ions. You also got the polyatomic ions down. Yeah. Phosphate is PO4. And but be able to recognize it in like this format here. So glycerol phosphate, that's something that's pretty important in different cells, and there's that phosphate group there. But again, emphasizing, notice all of the carbon chains, okay, and then you're swapping out those hydrogens for different functional groups. All right, methyl groups. That one should be pretty easy. We talked about that with like methane. Methyl group. Now it's CH3. Oh, I have a question for you. Why is it CH3? This says, it's too hard to see, dash CH3. Why is it not dash CH4? Actually, very important you understand that. It's kind of a subtle thing, but it ties back to the very, very first slides I showed you. Yes, go ahead.
Should I go?
Yeah, go ahead.
So because carbon can only make 4 bonds.
Yes, okay. But then why is it not 4 hydrogens?
Well, because if you want to connect to 4 hydrogens, you need 4 bonds. You can't have the extra bond.
We're trying to say is like we look at this example here. One of those 4 bonds is tied up right there, right? So that's why you can't have 4 hydrogens coming off of that. Yeah, when you see them show the CH3 group like this, I don't know if it's obvious enough, but that's just a shorthand way of this. Sometimes you'll see that where they're just trying to, it's like space considerations, so they don't have to like flare out those hydrogens like that. And These are the same ones. Yeah. Here's another list. Same ones. This is just like a simplified version. And I like this slide because one of the things you do need to know is polar versus nonpolar. Okay? All of these functional groups are polar except for methyl. Pretty simple. All of them polar except for methyl. But yeah, same shape here. What the R is referring to, R is referring to like a carbon chain. So this R could be like some like propane carbon chain that's being like, that's running over here or over there. Okay. All right. So anyways, again, not anything different other than make sure you recognize the polar and non-polar, all polar except for the methyl group. But just, I don't know, maybe a simpler way, different way to show them. Okay, I want to talk about ATP. So you should have heard of ATP before, you know, like throughout your schooling. ATP, you'll hear it described as like the battery of the cell. It's like the main energy source in the cell. ATP is referring to adenosine. So A is for adenosine. The T is for tri. Tri is referring to three. Think of a triangle. So tri what? 3 what? Phosphate. So adenosine and 3 phosphates. Okay? And now that we went over the phosphate functional group, the structure of ATP should make some more sense to you. Right? It shouldn't be quite as foreign. Okay? Anyways, ATP, remember the energy is stored in those phosphate bonds. So if you react it with water, you'll break off that ending phosphate, releasing a bunch of energy, and then you make ADP, adenosine diphosphate, diphosphate. And then that other phosphate is just floating around as what they call it, inorganic phosphate. Oh, question for you. Why do you think they call it inorganic phosphate as opposed to like organic phosphate? Think back to, I said organic compounds, like second slide. What was organic referring to? Yeah, it's not attached to like a carbon type chain. So hopefully you're starting to kind of, these words aren't being as intimidating anymore. You kind of have a basic kind of feel of what people are saying when they use those words. Okay. I'll make sure I said everything here. Yep. And again, how we release that energy stored in ATP is through releasing one of those phosphate groups. Okay, 3.2. Promise you it's a lot shorter. But polymers. Poly means many. Okay, like a polygon, many different polygons out there. Mono means one. So monomers make up your polymers. So those 4 biomolecules that I mentioned, like lipids, proteins, nucleic acids, carbohydrates, they all are polymers made of different monomers. So we'll go over those in the coming classes, but showing you how do you make a polymer from the monomers. So let's say we have a very short polymer. It's made of 1, 2, 3. Each of these spheres are each a monomer. If I wanted to link another monomer to this chain of 3 and make a chain of 4, I would want to do what's called a dehydration reaction. Dehydrate, if you're dehydrated, you need more water, right? So a dehydration reaction is removing a water. Meaning, what you're going to do is you're going to take what kind of group is that? OH group? What's that called?
Hydroxyl.
Yes, hydroxyl. What was another name for hydroxyl? No. Alcohol, yeah. So sometimes you'll hear it called hydroxyl or an alcohol group. So you have the OH group there. So that's where because water is made of OH, right? So you got the hydroxyl group there, the alcohol group there, coming from the monomer. And then the H comes from the ending part of the polymer. So you remove one of those waters, and then boom, you made a longer polymer. And you can keep doing this for however long you need to make that polymer chain. Okay? Now then, the opposite of building something is breaking something, right? So we can do a hydrolysis reaction. So hydro, you're hearing water again. Anybody remember what it means to lyse a cell? Like lysis? Cut, break, something like that. So hydrolysis, you're breaking down the polymer, and you're doing the opposite of dehydration. Now you're going to add back in a water to recreate that hydroxyl or alcohol group and that hydrogen. Okay, not too bad. Yeah, let me make sure I set everything here. Oh yeah, and those two reactions, yeah.
The other, the second one, the bottom picture, it had 1, 2, 3. Is that 4, or is that like a different?
This one? 1, 2, 3. Is this one 4?
Yeah.
Yes, yeah. Oh, I guess they don't call it 4 anymore because it's no longer connected to something. But yes, that was this one here. Yeah, good question. Yeah. So this last bullet point. Just a reminder that these reactions, those two reactions, are sped up by enzymes. So enzymes are proteins in your body that speed up all the chemical reactions your cells have to do. If you don't have enzymes in your body, you would not be able to do all the chemical reactions you need to do for your cell to stay functioning. Okay. And yeah, we have thousands of different macromolecules. So showed you some of those basic ways you can make these shapes. That gives you an idea of all the different kind of formations these molecules can take in your body. And those macromolecules, there's some basic, those basic 4 categories I gave you. All living things have those, but the specific kinds of lipids or specific kinds of proteins or carbohydrates, those differ from species to species. So there might be some unknown lipid that some random insect has, like there's like a species that's like the species of fish that can live in super cold waters. Their cell membranes, they're made of lipids, have these special lipids that don't freeze they freeze at a much lower temperature. So, anyways, basically, yeah, I want to run through this question because it's something you'd see on your test, and it's a good application question. It's kind of tricky. So they're asking how many molecules of water are needed to completely hydrolyze a polymer that is 10 monomers long. Okay, meaning this is 4 monomers long, but if I made it 10, right, I extended out that chain, how many waters would I have to add if I wanted to completely hydrolyze, completely break that down? And what a lot of people want to say is 10, right? It's 10 long, you need 10 to break it. But what you're forgetting is that you have an original water that remains. Like we kept with a shorter one. 1 water was used to split this, right? 1. A second water would be used to split that, 2. And a third water would be used to split that, 3. So only 3 waters would have been used to split that 4 polymer long chain. Does that make some sense? So in other words, however long of a polymer I give you, the number of waters you need to split it is always 1 behind, is what I'm getting at. Okay. And then, all right, last slide. Is this the same slide as, yeah, okay, yeah, it's a repeat. All right, so what you're going to do now for the rest of the class, you got the Mastering Bio. So work on the Mastering Bio, and then, yeah, once you're done with the Mastering Bio, then you can start reading for the next class, doing the reading guide. Maybe you could do the College Board, start working on that. But let me know if you got questions.